So, if you have any questions, these are the people to ask them. I should like to introduce the panel. If you wouldn't mind raising your hand and giving a wave to everyone so we know who we're talking about. First of all, we've got Professor Jeff Green. He's from the University of Sheffield here, and he's project lead and is currently teaching bacteria to eat grass. You might have thought the cows were doing it well enough, but if you want to ask more about that, you can get in touch with him. On the panel here, we also have Douglas Bruce-Kell. Douglas is a British biochemist and a professor of bioanalytical sciences from Manchester. So he's come a long way for this, across the Pennines. We've also got Dr. Robert Mecken, who's from Sheffield Uni, but will be going to Manchester shortly. You're particularly interested in the contexts and the processes of knowledge and how we disseminate these things in biology. We also have Dr. Esther Karan. You're a microbiologist from the University of Sheffield. She has spent the last couple of years studying the social life of bacteria. I'm looking forward to finding out what that means and how bacteria change their appearance according to the environment that they're in. And finally, we've got Dr. Alan Goddard. There we are. He's a senior lecturer in life sciences at Lincoln. He is currently involved in collaborations between industry and academia, exploring the potential of high-value chemicals produced by bacteria. Would you please give them a warm welcome? So we're going to try and keep this informal. We've called this session, well, we're talking about the unseen, and that's very, very fitting because for a start we don't often see what's going on in the labs, and we don't see what's going on at the scale of bacteria, things that are that small. If you have any questions at any point, please just raise your hand, shout at me or whatever, we'll try and get these people to answer. But tell you what, can we start with you, Jeff, because you were in the film there, and you say that the phrase you were talking about was like a factory. What makes a bacteria perfect as a factory?
I'm not certain what makes it perfect as a factory, but there are a number of features of bacteria that can be useful in terms of trying to make useful products, chemicals, biofuels, in a very sustainable way. So at the moment, much of what we see in the world relies upon fossil fuels, and those are a kind of non-renewable resource. They are finite, but we can use bacteria and yeast cells as well to produce chemicals that can sustain our lifestyles in a very sustainable way. So we can take feedstocks, which are currently waste products, and convert them into useful things. And so bacteria might not be the perfect cell factories, but we're certainly working towards making them better, and that's part of what CBMNet's trying to do.
So we're talking about trying to make the bacteria better. Now that involves what some people might find scary ideas such as genetic engineering and that kind of thing. Would anybody like to perhaps speak up for the benefits of using these technologies?
I will.
You'd like to give it a go?
Yeah, absolutely. I think genetic engineering is a really important technology, which I think is basically underutilized. Perhaps because of unfounded fears. Some of those fears may be well-founded, but mostly not. But in the context of industrial biotechnology and kind of producing chemicals and biofuels, then genetic engineering could be really useful, because you're kind of using these microbes as factories to produce useful products and not as food sources. So I can see the difference between kind of perhaps some uncertainty, some nervousness about genetically modified plants perhaps, but in the context of an industrial biotechnology in a cell factory, genetic engineering can have a great benefit. Sometimes called synthetic biology these days. So what we're trying to do is engineer these bacteria, yeast cells, to try and produce more product from less feedstock, and therefore benefit the whole of society.
Then I'd like to take this a little bit further, please. Tell us what is, actually, and also could you go into further perhaps what is actually involved in genetic engineering like this?
So you'll all be aware that Fleming discovered penicillin by a serendipitous circumstance in which he went on holiday and came back, and there was a plate of bacteria that this fungal spore had landed on, and all around the fungal spore the bacteria weren't growing. He realised that the fungal spore was making something that was killing them. The amount of penicillin as it was that this spore and this organism was capable of making was about 2 micrograms per litre, which is about a teaspoon of sugar in a swimming pool, if you want to feel free. That wasn't enough for the war effort as it then became, and in fact people had to reuse it by recrystallizing it out of urines and so forth during the war effort. But nowadays, to get to the sort of 100 grams a litre that the yields of industrial biotechnology making penicillin now took over 60 years. By contrast, when people worked out that they could make a model of the process, work out what was going on, decide which enzymes needed to be improved and boosted and so forth, in a recent project to make sustainable rubber using microorganisms, it took them 6 months, because essentially the bacteria that they were going to use to do it had all of the enzymes except one. Then they took this extra enzyme that they needed from a poplar tree, which made little bits of isobutene, which is what's in natural rubber, and in 6 months they got it up to 60 grams a litre. So the power of the techniques comes from the fact that you actually know what you're doing.
Yes. And of course, one of the reasons why we're doing this artwork, this mural, is to talk about actually trying to get the chemicals out of the cells. So all these people work in some way on the boundaries of these cells. Would you actually like to tell us a little bit more about that? Can I come to you, please, Esther, because I just loved your biog of saying that you're studying the social life of bacteria as well.
Yeah.
What does it mean to be studying the social life, and how is that useful for making these technologies work? That wasn't her, that was a microphone. It's okay.
Yeah. So when I was studying in university, I tended to think of bacteria as very lonely organisms. We are taught that they are single-celled, they live on their own, they cause disease. So this is the kind of opinion that we have. But when I came in to do my research, I found that bacteria are not like that at all. They actually rely on each other, and they can form little groups. So they rely on their friends almost in order to overcome things like environmental stresses. So if it becomes too hot or too cold or even too salty, what happens is that these bacteria huddle together in little groups. They try and make most out of this stressful environment that they are put in. So, so they're quite social, very different from what we're all taught at school.
And when we talk about social in this case, it is that they're doing it chemically across these membranes, I guess.
Yes, so one way that they recognize that there are others like them around themselves is that they produce these molecules, these chemical molecules called form sensing molecules, and they produce that from within the cell and secrete it outside. And once there are enough molecules, they talk to each other, and they start producing little fingers and hands on the surface that they hold onto each other. And I'd like to think of it as if they're giving each other a hug. So they hug each other and sit in this little social colony, if I can call it. And I've been studying that really.
How do you feel knowing that we're all covered in bacteria holding hands at this very minute? How lovely. Well, if we're talking about exporting things across these membranes, can I come to you, Alan, for a second? Because some of these things that we're exporting, and particularly when we genetically modify them, we're talking about the phrase we have in your biog here is high-value chemicals. Tell us more. Like, are we going to get gold? What are we talking about here?
There are bacteria which will eat metals, for example. So you could, in theory, there's something called bioremediation, where if you've got a spill of heavy metals or oil or something, you can use bacteria to basically eat those, and then you can gather the bacteria and extract the products from those. But my research is more based around kind of the more fundamental things we use day to day. So biofuels is one of the examples. So you all know that you put petrol or diesel into your car. You've probably all heard of bioethanol. So you can use ethanol in a mixture to power your car. And ethanol is actually a really inefficient way of powering your car. Something like butanol is far better. The problem we have is that ethanol, you can make it in yeast, and you can make it up to about 18%. So if you've ever had Japanese sake, which comes from fermented rice, it gets about 18% alcohol. Butanol, you can only get to about 2%, and the cost of making all these fuels is the extraction process. So when the bacteria have made them, got them outside the cell, you then have to get them out of the slurry that the bacteria are in. So ethanol is really cheap, butanol is really expensive. And butanol is toxic to these cells, as you'd expect. It's quite a nasty chemical. So we're working with some companies to try and optimise how the cells make the butanol, how they export it, so then the extraction becomes cheaper, so then the fuel you buy becomes cheaper. So biobutanol, they've just opened a plant in America as a test case to show it's commercially viable, but it always competes with the price of oil. And the price of oil is currently very low, so what we want to do is make the butanol far cheaper to produce, which makes it far more commercially viable, and it's a green fuel as opposed to fossil fuel.
So this is the kind of research that's actually even happening here right now as well.
So I'm working with a company based in Oxfordshire who are working to improve this process, and we've got a few different strands going on, one of which is genetic modification, one of which is looking at the membranes and the composition of the membranes around the cells to make them more resistant.
Now, I'm coming over to you, Robert, because your interest in this is partly getting all biologists like this from different disciplines to talk and also to listen to the kind of problems, the ethics behind these sort of things.
Yeah, I mean, I'm a sociologist, so I look at how mostly how biologists work and how they present their science and how their science is, or various scientific disciplines are shaped because of political reasons or material or organisational concerns.
So do biologists work? That's the real question. I hope
they work very well together, I think. They often hug one another. But so one of the things that's interested me, for example, is the idea of cells as factories. Like that's a particular kind of analogy or metaphor that's quite prevalent in synthetic biology and in biotechnology in general. And I think it's interesting to characterise that with genetic modification, where there was a big concern there, the idea that the public were concerned about cross-pollination and the idea that the genetically modified crops could escape and were not controlled. And now we're presented with factories as an example. By the way, I should say they've been asked to be radical and provocative. So factories, so we're thinking of a car factory was the analogy that we were given, which sort of presents the science as safe, as mundane. We're producing chemicals, compounds that already exist, and it's implicitly beneficial to our society. I mean, car manufacture doesn't come just as a benefit, right? It comes with all kinds of other pollutions and accidents and so on. So that's the kind of question when we're talking about analogy and how we present our science. That's the kind of things that I'm interested in at times.
Can you give us a good example then of, is there places where it has gone wrong in the past, and have we learned from it?
In terms of, how do you mean?
So, I suppose you were saying that you were very interested in how, say, people are afraid of some of these technologies because they are very often presented as just being good. Is there any examples of where some of this genetic modification has gone wrong so far?
I don't come at the question the way that you've come at it, I think. I'm not interested in whether things are right or wrong, or they've done well or badly. I'm interested in how things work. So you don't look at a photosynthetic tree and say it's doing photosynthesis wrong. Yeah. You look at society. So I look at society and I look at how it works. There is an example of synthetic biology that Jeff mentioned before called the Artemisinin Project, which was set up in 2003 to produce anti-malarial drugs using fermented yeast to produce them cheaply for the world's poorest. That was the original promise, but the factory was sold earlier this year, so it was scaled up with Sanofi Aventis, a big pharma. There was a huge investment from Bill and Melinda Gates Foundation of $50 million. But over this period of time, the synthetic biology promise of delivering cheap drugs for the world's poorest has sort of been eroded and in fact not realised because they've had to sell the factory because there weren't the market forces weren't conducive to producing antimalarials in this particular way.
So what we're really seeing here is that biology does not exist in isolation. It is interacting with the real world and economics and things like that here.
Absolutely. Yeah.
Well, I could have a ton more questions for these guys. Before I get another ride at them, if anybody has any, please raise your hand. We've got lots of microphones around here. We want to see the kind of things people in Sheffield want to hear as well. Go for it.
So, by and large, that's not been the case. There were early proposals, for instance, to make genetically engineered proteins in the milk or eggs of farm animals. It didn't work, basically, so nobody's doing that. There are much more thoughts of making plant-derived chemicals. So there's a famous example, so-called purple tomatoes. People at the John Innes Centre in Norwich developed ways to increase the amount of chemicals called anthocyanins that are in stuff like beetroot in these tomatoes, so they were purple rather than red, and they fed these things to mice that were prone to cancer and put the equivalent of 17 human years, 40 days on the life of these mice. So clearly the things were good for you, but you're not allowed to grow them in this country. But currently they would be grown as tomatoes, not as bacterial versions of purple organisms. And while the legislation is such preventing it, it's probably going to happen in Canada, so we will have to re-import the things to us so that we can have the benefits of living longer by eating healthy purple tomatoes. But that's the way it is. But that's an example. So some things make sense to be made in organisms other than bacteria and yeast and fungi. For other things, perhaps less so. They're more sensibly made in the organisms they're normally made in.
I think my favourite example of one of the ones made in organisms, if you haven't heard of this, please look it up, it's great fun. It's called spider goat. It was a goat which would make spider silk protein in its milk. They called it silk milk. Is that not incredible? It always makes me imagine that milking it, you get something like cheese strings out the other end. Really rather vicious. Boy, hands up. Who would like to try these purple tomatoes if they were given a chance? And who thinks it's repellent and wrong and possibly taste awful? Hands up if you're indifferent. Indifferent people don't often put their hands up, actually. It's part of being indifferent. But, you know, well done. Have we got any other questions from the floor?
I'd like to also feed into that question if possible.
Yes.
There's another reason why I don't personally see this technology moving towards animals and plants, because plants, for instance, have a season where they might be able to produce the protein that we have engineered either in their flowers or in their fruits, and that involves a waiting period, and also the same in animals. So essentially, when you're thinking of moving the genetically engineered protein from a microorganism to a plant or an animal, what we are saying is that we have to wait longer to get to that protein that we are interested in, rather than getting it within 20 minutes, say, from a bacterium. So that's the reason why I think that this technology won't go in that direction.
Yeah, I think those are good points, and I think also we've still got a lot to learn at the microorganism level before there's any large-scale movement into higher organisms. And you could look at, say, animal cells rather than animals. So there are kind of various cell lines that are massively beneficial, and many therapeutic proteins that are used in all kinds of treatments, especially for cancers. Antibodies are made by animal cells in culture. So they're not animals, but they are animal cells that have been given the ability, or have the ability, to propagate in a similar, not exactly the same, but in a similar sort of way to a bacterial or a yeast culture. So I think that we still have a lot to learn at the microbial level, and as has already been said, there are lots of advantages in kind of using these microbes, because you don't have things like, what's the season or maturity times. You can do things rapidly, which I guess kind of links back to something that was said earlier on. Partly because of the relative simplicity of microorganisms, and I think we all agree that they're not simple overall, but the relative simplicity allows you to maybe understand more, and therefore be a bit more rational about your progress, so things don't take decades, they can potentially take months.
It does seem that there is legislation around these things as well, though. Like, it seems to be tricky, and you have to get different hoops to jump through for each different type of organism. I'm just thinking that I was aware in the news, you may have heard it a couple of, maybe about a year and a half ago, there were some people on Kickstarter trying to genetically engineer plants to glow in the dark, so we wouldn't need as many streetlights. Their intentions were well-founded, they were very interesting as a way of cutting down for the need of electricity. But yeah, it was quashed pretty heavily. I believe it was stopped. Does anybody know any more about this? This might be... No?
The related thing I know about is the only GM organism you can actually buy, the only GM animal you can buy, called glowfish, and they're made in California, and they're basically zebrafish, which people have put fluorescent proteins into, and you can buy all the different spectrum of colours of fluorescent fish now and buy them and swim around your fish tank. I think it's restricted to America, but the genetic legislation in this country is pretty much watertight, to be honest, because I sit on our university internal GM board, and that's pretty watertight. And so when you go a bigger picture, even outside of university, the regulatory affairs with GM crops and GM animals is huge.
Terrific. We've got another question over there. Would you mind getting a microphone to that gent?
Radiation causes mutations. So if you go out in the sun and you expose yourself to loads of UV light, you get a mutation in your DNA which can cause skin cancer or whatever. Radiation is a very inefficient way of introducing new things into organisms, so you can remove things or change the function of what's there already, but you're highly likely to introduce something completely new. So you're not going to get, if you've got a load of zebrafish and exposes radiation till the end of time, you'll probably never get one that glows in the dark. It's such a targeted new thing. But you could make it have different additional features. And one of the, I'm going to say old-fashioned, as in I did it in my PhD, ways of mutating bacteria is just expose them to UV light and see what you get at the end of it and select based on what you want. So it's a very quick, efficient way to find things, but now we've got whole genome sequencing and we know what's going on with each organism, it's kind of passé now, I think.
So nuclear does work, but we've just improved and moved on from it. We have better techniques nowadays. Is there any more from the floor?
So these molecules that I talked about, quorum sensing molecules, it's a type of a language that the bacteria use to communicate amongst themselves, but they also have different versions of these chemical molecules that they can use to communicate, for instance, with archaea or with other cyanobacteria or even yeast. And so there is literature that says that yeast and bacteria now communicate with each other. And now what the direction that the research is moving in is that we are trying to see if there is a third kind of a language. So one language to talk amongst themselves, another language to talk amongst other microorganisms. And there's also, now we are trying to see if there is a third language that allows them to talk to higher organisms like humans, for instance. So it's very much at the forefront.
What you say bacteria talking to humans. What does a bacteria say to a human?
It says, give me more iron, for instance.
Please elaborate. That's a good story. Yeah, tell us more.
Taking that further, there's an example based exactly on these things where other organisms talk to the bacteria and say, I don't want you. And in this case, they make a molecule that looks a bit like, these are seaweeds, and they make a molecule that looks a bit like these quorum sensing molecules, and the bacteria think, oh, I don't like to be here. And it was discovered that they don't, you will know the story, won't grow on these seaweeds, which of course is good for the seaweeds. So they've essentially hijacked the language of the bacteria to interfere with them to get rid of them.
So does that answer your question okay? Because this is a very fascinating side of science, because if we can learn these languages, I suppose the next step is that we can talk back, as it were.
Absolutely, and control what response that we want the bacteria to give us, definitely.
It's blue sky thinking. What would you like to say in these situations? What is the hope that you have for these communications?
Well, can I
Of course,
just chip in?
yeah.
I was just going to mention that if the title was The Fear of the Unseen, and in some ways biofilms are something to be slightly scared of, yeah. So all of these kind of communication models that molecules that are being exchanged within these communities of bacteria are being studied by many people with the hope that if you can interfere with that communication process, so the bacteria can't talk to each other anymore, that could be a source of new types of treatments for various diseases, which are dependent upon the bacteria getting together and acting as a community. So there are many types of infection where an individual bacterium on its own, acting on its own, cannot cause a disease. But when they all get together and act as a group, producing toxins and things that way, if you will, if you could interfere with that by blocking these communication channels, if you like, that could be a great step forward. So there's lots of research going on in that area. Do you think that's true?
Yes, I think it's absolutely true, actually. And such treatments have already started because now that we know that bacteria can almost hug or hold hands and to form this group and then mount an attack, we can interfere with that holding of hands or of the bacteria getting into little groups. So one of the cases in point is that when people have cystic fibrosis, the reason why it flares up or it becomes worse is because you have bacteria called Pseudomonas that infect them and form these little groups and mount an attack. So now the therapy for cystic fibrosis includes inhalation of an enzyme called DNase, because these little hands that bacteria use to hold each other, that research has found out is made up of DNA, so extracellular DNA. So now patients are being prescribed that they inhale this protein or this enzyme that breaks down DNA, and then as a result these little bacterial groups are sort of scattered now, and the existing therapeutics can actually treat them. So it's actually in use now.
We're stopping bacteria ganging up on us. What a wonderful idea. Actually, there was a couple of terms there actually I'd love you to tell us a wee bit more about. I heard biofilms. Can you tell us more what those are? And can you tell us, what is actually involved in a hug from a bacteria, and do I want one when I feel sad?
Definitely. A biofilm is exactly what the word says. It's a living film, therefore it is a biofilm. And the way that these films form is that bacteria produce sticky molecules. So basically they produce glue, and they secrete them out of their cell membrane into the extracellular environment, and then use that as basically superglue. Superglue is what I'm looking for. They use it as superglue to stick onto surfaces. So, and they form layers, and that becomes a living film.
Yeah, they're really complex structures. They can be communities of all kinds of bacteria and other organisms in there, and they can have channels in them, so nutrients can flow through them. And many of the substances that they excrete to kind of attach to surfaces and form the biofilms, they can be polymers of sugar molecules, some of which are used in face creams and all kinds of things to kind of make us feel better or look better. They can include DNA, which is a very important part of a biofilm, and also proteins. And so the idea of kind of the binary link, that's kind of one bacteria held close to another bacterium, it's more complex than that, because they all live inside this great kind of matrix of polysaccharides and DNA and protein, and so they're interacting in a more complex way than a kind of one-to-one
Does that make sense?
relationship. Yeah.
Brilliant. Are we getting more?
That's a fascinating area, because so far I've talked about how harmful they are, as in cystic fibrosis, they worsen the disease. But then going back to my previous statement that I said, huddling into these little groups help the bacteria to overcome the stress that they might face in a situation. So at the moment there is a project that I'm working on that is looking at all these clusters of bacteria that is helping to treat wastewater, for instance. So all the toxic molecules that are in wastewater can be degraded in a biological sense if we use these little colonies, group of friends.
Does anybody have anything else to add? Any examples they particularly like?
Yeah, I think there are many potential applications of biofilms. You know, we hear about the bad ones where something might be attached to a catheter and things, but they can be really useful and can be developed in a way to kind of in bioremediation, in kind of taking the toxic elements out of, for example, wastewaters and other waste streams. So that's really a good application of biofilms. But then biofilms exist in healthy situations as well, you know. So for every one of us, there are 10 times, at least 10 times more bacterial cells in and on me than there are human cells. I am mostly bacteria, and so are you. And they perform a really useful role because you're healthy, and they help to protect you from the more harmful bacteria, and as part of doing that they're colonizing surfaces, be it your skin or your intestine, and forming biofilms is all part of that. So they are really important things.
Just like to make a small point about promises and biotechnology, because biotechnology often makes a lot of promises about the goods that it will deliver, and at the moment we're seeing a lot of sustainability and the recycling of waste, and this what's called the circular economy, where we'll reprocess waste issues. That's kind of how it's been positioned at the moment, but we know from the Human Genome Project, which was supposed to deliver huge health benefits for us, that in fact what we learned as much as anything is that the genome is much more complicated and much more reactive to its environment than we previously thought it was. So when biotechnology is making promises, we need to just treat them with a little bit of caution in terms of their potential.
Is there anything you have particular hope or skepticism for?
I don't personally have any issue. I'm looking at a historical case study of the Human Genome Project and applying that to what we're talking about today.
Cool.
So, well, it was a broader question about where does innovation and creativity come from, and one of them is knowing what's out there in the first place. So being widely read is a sine qua non for having a creative insight. A nice example I'll take, slightly less, it is microbial. So Procter & Gamble, who do face creams, discovered that the people who make sake that was mentioned earlier, sake starts life as a kind of yeasty, doughy product, and the people who were putting their hands into it like this, aged in their 70s, had skin like babies' bottoms. And then folk realised that actually there must be something in this stuff that was beneficial from this point of view. And if you've ever heard of SK2, spelled 1-1, this is an expensive product flogged by Procter & Gamble, which does actually work to preserve your skin in a nice youthful way, but it's just basically nutrients that are in there. But it was a serendipitous observation again, someone with their eyes open thinking, hey, here's something that is unexpected, and let me try and find a scientific explanation to it. And I could give you a long list after we've had the formal panel, but others can join in.
Ask him later for more serendipity. I'll be Owen.
I think for me, a lot of the exciting things now is the industrial biotech type of things where we've got hundreds of years of basic scientific knowledge and we're now starting to apply it to real world problems. So that's kind of the journey I've been on personally, where my PhD was very fundamental science. When I was a postdoc, it was very much, let's look at this one tiny element of something and find out how it works in huge amounts of detail. And then becoming involved more with industrial applications makes you look at the bigger picture and try and apply what you know to a different problem, which is the thing that excites me. Some people are excited by their one little protein for their entire career, whereas I kind of like the techniques and the things that I know and applying them to bigger real world problems.
You could tell something, please?
So the questioner asked us to look into the future. You don't have to look very far into the future. If you look at Pokemon Go, imagine an app in which you're walking around with your smartphone and your Google Glass, and it's telling you all about the fantastic sciencey things that are going on all around you, because it'll be able to do that. And that's not very far into the future. So people writing apps of this type is clearly going to have a huge impact on benefiting anyone's interests and facts in public science. If people are interested in finding a wretched Pokemon, you'd think they'd be interested in what's going to be exciting wandering around wherever they are in a city or wherever it be. So I'd be very optimistic personally about that sort of thing.
And last word with Robert is, this is kind of your area.
I'm an advocate for engagement and certainly for democracy, but one of the things that I would say is that we know from social studies that engagement and communication helps people to understand the science, but also to raise more nuanced resistance to science as well. So it's not just about public acceptance in that way. And in this way, when we're talking about goods and public goods, I ask the question, well, whose good is it? If we're talking about industrial biotechnology, who's going to reap the benefits of industrial biotechnology? Is it big companies, or is it the general public?
What a kind of hard question to end it on. Go off, do more research. I'm sure you guys don't mind hanging around if these people want to. Yeah. I've socially thrusted upon you now, if you don't mind hanging around.
Yeah.
So these people can ask you more questions if they like. It's been lovely talking to you. This is only one small part of the Sheffield Festival of the Mind. Please go off and check out everything else. Before you do go, please give a huge warm round of applause. We've had Professor Jeff Green, we've had Jen who's helped us organise the whole thing. There's been Professor Douglas Kell, we've had Robert Meckin and Esther and Alan as well. Give them a huge round of applause.